Hello everyone, today we're going to be presenting on Toxoplasma gondii, which is one of the most widespread but often overlooked parasites in the world. It's known as the silent killer because most infected people never realize that they even have it, yet it can cause serious and sometimes life-threatening diseases in vulnerable groups. Our goal today is to walk you through the biology, transmission, human disease, and environmental impacts of this parasite.
So what is T. gondii? Toxoplasma gondii is a protozoan parasite that is capable of infecting almost all warm-blooded animals. What makes it very unique is that it can live inside nearly any nucleated cell of the body. On this slide, you see its three main forms: oocysts, tachyzoites, and bradyzoites. Tachyzoites spread during acute infection, while bradyzoites form long-lasting cysts. Cats are the parasite's definitive host. These can be domestic or wild felines, and that means that cats are the only animals where T. gondii can reproduce sexually and shed oocysts into the environment. Even though most infected people never show symptoms, the parasite does become dangerous for pregnant women where it can cross the placenta and also for immunocompromised patients where it can reactivate itself.
Okay, so this slide is going over the life cycle and transmission. Cats become infected whenever they eat prey or raw meat containing tissue cysts. Inside the cat's intestine, the parasite undergoes sexual reproduction and the cat begins shedding millions of oocysts. These oocysts contaminate soil, produce, water, and even coastal environments, and they will survive for months, even up to a year in most conditions. Humans and other animals become infected in the three major ways. Firstly, by ingesting oocysts from contaminated soil or water, by eating undercooked meat containing bradyzoid cysts, or through congenital transmission, which is from pregnant women to a fetus.
So this slide is about the human disease caused by T. gondii called toxoplasmosis. Toxoplasmosis simply means infection with T. gondii. For most healthy people, the infection does remain silent and they usually don't show symptoms, but certain groups are at high risk. For example, pregnant individuals may pass the parasite to their fetus, where it can cause severe developmental issues. Immunocompromised patients, such as those with HIV or AIDS, or also on chemotherapy, can experience reactivated infection, where latent cysts suddenly multiply. We divide infections into acute and severe or reactivated infections. Acute infections are often so mild that they go unnoticed, whereas reactivated infections can become very life-threatening.
So this slide just expands a little bit more on the immunocompromised individuals. Patients that have toxoplasmosis gondii can invade, the parasite can invade the brain, eyes, lungs, or muscles. Neurological symptoms can include headaches, seizures, confusion, and poor coordination. And on imaging results, these patients can typically show ring-enhancing lesions in the brain, which are the circular white areas that represent inflammation and active parasite replication.
This slide summarizes treatment and prevention strategies. The standard treatment for serious infection is pyrimethamine, sulfadiazine, and leucovorin, which is a combination that targets the parasite while also preventing bone marrow toxicity. For the patients who can't tolerate sulfa medications, pyrimethamine and clindamycin are used. Pregnant individuals are usually treated with different medications. Spiramycin is used early in pregnancy to decrease the chance of fetal infection. Prevention includes thoroughly cooking meat, washing produce, avoiding untreated water, and avoiding cat litter if pregnant or immunocompromised. Prevention focuses on managing cat feces and protecting livestock feed from contamination.
Okay, so this slide just breaks down the diagnostic process into four steps: serology, PCR imaging, and amniotic fluid testing. These steps kind of help clinicians confirm infection, assess severity, and determine risk for food safety.
This slide goes more into depth on the treatment aspect. Acute therapy typically lasts 4 to 6 weeks. The duration of this therapy is important because the parasite replicates quickly and must be suppressed long enough to stop the disease from progressing. Ocular toxoplasmosis often requires even longer treatment, and immunocompromised patients usually need lifelong suppressive therapy to prevent the infection from reactivating.
This slide highlights both human and environmental prevention. Cooking meat above 160 degrees Fahrenheit or 71 degrees Celsius kills tissue cysts. Washing produce removes oocysts from the soil, and drinking clean water can prevent waterborne outbreaks. On the environmental side, preventing cats from hunting wildlife, disposing of litter properly, and protecting livestock feed and water reduces contamination.
This slide discusses the agricultural impact of T. gondii infections, as farm animals, especially sheep and goats, are highly vulnerable. T. gondii infections in these types of livestock cause reproductive losses, including abortion and stillbirth, and cause major economic concerns. Meat quality also decreases, and sales go down when cysts are detected. Our primary research article, Stelzer et al., emphasizes that the economic burden is vastly underestimated because surveillance in livestock is very inconsistent across different countries.
Okay, so just piggybacking off of the environmental impacts, oocysts from cat feces contaminate soil, rivers, and oceans. They can survive up to 12 to 18 months in moist environments and are extremely resilient. This slide mentions die-offs in marine mammals. Sea otters, dolphins, and seals especially can be affected from runoff carrying contaminated particles into coastal waters.
This slide discusses the global impact and prevalence of T. gondii infections. About one-third of the world's population has been exposed to T. gondii. That alone makes it one of the most successful parasites ever recorded. The prevalence of T. gondii infections varies enormously, as some regions have as low as 10% prevalence, while others reach over 80%. Warm, humid climates and regions with more cat to environment interaction tend to have higher rates. And this is seen on this slide where this figure from one of our references, Galvan Ramirez et al., maps the global seroprevalence. Seroprevalence is basically the level of a pathogen in a population, which is measured in blood serum. Darker, more saturated regions show higher infection rates. You can see that there are high concentrations in Latin America, Africa, the Middle East, and also parts of Asia, whereas lower prevalence is seen in Northern Europe, North America, and China.
This slide summarizes our primary research article. Stelzer et al. Examined infection in farm animals and found that outdoor farming systems, poor rodent control, and unrestricted cat access are the major risk factors. That likely shows farm animals, environments, or livestock, reinforcing the connection between animal management and infection risk. A major contribution of the paper is the economic analysis showing how toxoplasmosis leads to abortions, stillbirths, reduced productivity, and major financial losses in agriculture.
This slide highlights the key results of the article just mentioned, where high infection rates in sheep, goats, and pigs occur, as well as outdoor farming increasing exposure, and rodent control and cat presence being major factors. Also, many infections are underestimated due to inconsistent testing.
Okay, so why does this research matter? Research on T. gondii matters because it is one of the most prevalent infections worldwide. It affects public health, animal health, agriculture, and the environment. For pregnant individuals, congenital toxoplasmosis can cause lifelong disabilities. For immunocompromised people, it can be fatal. For farmers, it can create economic strain, and for the environment, it threatens wildlife, including marine mammals. Improving surveillance, treatments, vaccines, and prevention strategies is essential for reducing the global burden of toxoplasmosis.
And these are our references that we used in this presentation.